The following is a selected video from masterthecontent.com, where you will find an extensive video library of lectures for a variety of standardized admission tests. We offer over 600 hours of detailed video lectures for a multitude of standardized tests. Use our interactive in-lecture table of contents to find specific topics of interest. Work through numerous in-lecture examples to help you internalize concepts. To learn more, visit masterthecontent.com. Your career, our passion.
Bacterial cell wall structure. Now, the cell wall is obviously responsible for the shape of the bacterial cell. The cell wall is a complex, semi-rigid structure that actually helps determine the shape of the cell. Now, underneath that cell wall, we'll have our regular cell or plasma membrane. Now, why the cell wall is important is, for instance, under conditions of desiccation under very dry or osmotically intolerable conditions, what the cell wall will do, it'll prevent the cell from either losing too much solute to the environment or gaining too much such that it bursts. So basically what the cell wall helps maintain is the structural integrity of the bacterial cell. It ensures that it doesn't gain too much solute or doesn't have a solute concentration that would cause it to burst. We call that lysis. And it also ensures that if it's in very hypertonic solutions, a very hypertonic environment, that it doesn't actually shrink and die for that reason as well. Now, so that's the major function of the bacterial cell wall, to prevent rupture because of turgidity. It also helps maintain the shape, like I said. And one additional function that is also important is it's an anchor point. It serves as an anchor point for the attachment of motility structures like flagella or like cilia. So those motility structures that the microorganisms or that the bacterial cell uses to get around actually attach to it in the cell wall. And I'll go over that as well in the coming slides. Now, what I want to do is briefly talk about the composition of the cell wall. Now, I talked about peptidoglycan a lot at the beginning of this lecture. Now we're actually going to examine its composition. So a bacterial cell, or all the bacterial cell wall, is composed of this compound I call peptidoglycan. So peptidoglycan is a polymer. It's actually composed or comprised of a series of repeating monosaccharide units, repeating disaccharide units, that are cross-linked or held together by shorter polypeptides, and they're held together in such a way that they form a protective lattice. So the layer of peptidoglycan has a very important structural function in cell walls. It gives it structural and mechanical strength and support. So it provides rigid support, and at the same time, while providing that rigid support, it still allows all those substances to pass across the cell wall. So it's fully permeable. It maintains solute permeability. So it doesn't affect the permeability of the cell, but it increases its structural strength, its mechanical strength. Now, so those disaccharides I told you, I just mentioned. So we said peptidoglycan is composed of repeating disaccharide units. It's a polymer. So let's talk about those individual disaccharide units. There's two main kinds that you will find. The first one is N-acetylglucosamine, sometimes abbreviated as NAG, or N-acetylmuramic acid, abbreviated as NAM. So those are the two main disaccharides you will find that polymerize to form peptidoglycan. Now, the components of peptidoglycan are actually assembled within the bacterial cell wall. So what's going to happen is NAG or NAM will find its way or will be transported to the bacterial cell wall where it will actually be assembled into that peptidoglycan layer. Now, these alternating N-acetylmuramic acid and N-acetylglucosamine molecules are actually linked in what are rows. Now, I'm going to give you a diagram to illustrate what I'm trying to talk about before I actually get into much more detail. Here is what we have. We have a gram-positive cell wall. Now, this is the peptidoglycan I was talking about. As you can see, we have those rows of those disaccharide molecules arranged in the cell wall. Now, what's going to happen after that is that we will have shorter proteins or peptide links coming in, linking those rods or those chains, those polymers together to form a very structurally rigid layer, to form a very mechanically sound layer that confers mechanical support to the cell wall. So That's what it looks like. That gives you a diagrammatic representation of what I'm talking about. Now, so let's move on.